get started. So today's topic is on metabolism. There was a part one that another person on our team, Ashley, did. So if that's something you want, when I send you the post email tomorrow morning, you can reply to that asking for that session also. There's slides and recording of it. So that covered anaerobic respiration and like glycolysis, gluconeogenesis, and that kind of stuff. So today we're going to be talking about the aerobic side of it. And then if you guys haven't done a session with me, my name is Pranava. So just a couple announcements before we get started. As always, if you can afford it, donate to our PayPal. So hopefully we can reach more students and then keep up our website and a couple other expenses we have. But no pressure, especially if it's like a pressure on you to do that. We also do have a Facebook group that's up and running, so you can join that if that's something that motivates you while you study. And then lastly, if there's any questions about whatever we discussed today or just anything in general about the MCAT that we might be able to help you with, email us at mcat101questions@gmail.com and we'll get back to you. So today's session, you can unmute yourself at any time and interrupt me, put it in the chat. And as always, if you see anything that I say wrong, feel free to correct me, or if you think it's wrong and it's not, it's totally fine. Just try to be as interactive as possible, because when you're actually engaging, that's probably when you learn best. So yeah, let's just get into it. So again, I was talking about the overall metabolism overview. So this is all of it. Ashley covered anaerobic respiration, so glycolysis, fermentation, and then gluconeogenesis, glycolysis, and I think glycogen metabolism. Today we're going to be talking about these three: pyruvate dehydrogenase complex, Krebs cycle, also known as the citric acid cycle, as well as the electron transport chain. So if you look at this picture, it just orients you as to where in the cell these processes occur. So glycolysis and most anaerobic things take place in the cytosol. Everything we're going to be talking about today takes place in some part of the mitochondria. So this is the first part. So if you remembered back to glycolysis, the end product of glycolysis is pyruvate, which is this 3-carbon compound. But to utilize this and move on into the Krebs cycle, what needs to happen first is that acetyl-CoA needs to be made. And acetyl-CoA is this 2-carbon compound. And this is a compound that goes directly into the Krebs cycle. So after glycolysis, before entering the Krebs cycle, this process needs to occur in the pyruvate dehydrogenase complex to yield us this acetyl-CoA. So this is called a oxidative decarboxylation reaction. If you see what's highlighted in the pink, that 1 carbon of pyruvate is lost as carbon dioxide. So that's how your 3-carbon compound of pyruvate becomes a 2-carbon compound of acetyl-CoA. So that's the decarboxylation part of it. And then the oxidative part of it is that you can think of it as like this thiol-CoA group getting added on. That's the oxidative side of it. Another way to think of that is that you have a NADH forming, so this NAD plus is being reduced. So to balance that out, the pyruvate is the one that's getting oxidized. This is kind of a question that comes up often, like framed like this. They might just give you like one reaction, like pyruvate, and show you acetyl-CoA without giving you the names. And these structures aren't something you would have memorized. So when they just give you this reaction, you should be able to tell by seeing that, okay, here's your NAD plus that's getting reduced into NADH. That must mean that pyruvate to acetyl-CoA is getting oxidized, because your redox reactions are always happening in such a way that when one of your compounds is getting reduced, the other one should get oxidized. So we talked about pyruvate and NAD+. Your other reactant is this CoA with the thiol group, the SH group. So that gets added on right onto the second carbon of pyruvate. And then I have here that it requires oxygen. So again, this is all an aerobic process. So this would require oxygen. If oxygen is not available, this pyruvate would instead be fermented. Um, and the location is the mitochondrial matrix, which, if you don't remember, it's the very inner part of the mitochondria. So not either of the two membranes, but the lumen, so the portion inside the inner membrane. Um, the only other thing that's on this slide that you would need to know is these four cofactors. So cofactors are things that are necessary for the enzyme to function properly. Um, I don't know that you need to memorize this, but it's not that big a deal, so you might as well go ahead and memorize it, because I think, I'm not completely sure, but I think I've seen a question where it's like, which like enzyme complex requires like lipoleic acid, which is another name for lipoate. So stuff like that might be good to know. Yeah, sorry, I don't know if I misspoke, but in the chat, somebody asked if it requires oxygen, wouldn't it be aerobic? Yeah, that's right. I'm sorry if I misspoke. It requires oxygen, so it's aerobic. This is all part of aerobic respiration. Thank you for correcting me. So yeah, I would just recommend that you go ahead and memorize most of this. So you don't necessarily need to memorize the structures of pyruvate and acetyl-CoA, because that'll eventually get too confusing once we talk about a variety of different compounds that come up. But know the name, know that pyruvate is getting oxidized, know that this process reduces NAD plus to form NADH, and that it yields a carbon dioxide, and that these cofactors are needed. Another question in the chat. Yeah. So where does oxygen fit in here? Yeah, it's not like a direct reactant in this, but it's when you don't have oxygen, the cell has other mechanisms to use the pyruvate. So it's kind of like a balancing game. I don't know the exact details of the mechanisms beyond what's on this slide, and you don't need to either. But it's just know that when the cell doesn't have oxygen available, the pyruvate would not go through the pyruvate dehydrogenase complex to form acetyl-CoA. It'll instead either go through alcohol fermentation or lactate fermentation. Is that okay, Karen? And then with delta G, this, if you remember, is the Gibbs free energy of reaction. So the negative delta G just explains that it's a exergonic reaction, so that it releases energy. You don't need to input energy. This isn't anything that'll come up. It just happened to be on this picture, but you don't need to worry too much about that. But okay. And then, yeah, Karen, it's oxygen is directly reduced in the electron transport chain to form hydrogen or water. That's correct, but just that it needs to, even for this very first step to occur, you need to be in an aerobic environment for the cell to choose to take pyruvate down this path, I guess. You're welcome. And this is just another different picture that I found to kind of represent this in a different way. So this is a complicated enzyme complex as opposed to being one enzyme. So we generally just call it the pyruvate dehydrogenase complex, but that's like complex 1. There's another one, acyltransferase, and then this dehydrogenase. You don't need to know the details of that either. I just wanted to let you know that this is a bigger complex. Yeah, and then only other thing that needs to be mentioned on this slide is that remember that because one acetyl-CoA in this process yields 1 NADH and 1 carbon dioxide. For every 1 glucose, that would mean 2 NADH and 2 carbon dioxide because every glucose, which is a 6-carbon molecule, yields 2 different pyruvates. And pyruvate dehydrogenase is an oxidative reductase enzyme, which fits in with what I was saying about like pyruvate being oxidized and NAD plus being reduced. So it's a redox reaction, and the enzyme is an oxidative reductase.
Could you please repeat about the 1 acetyl-CoA is equal to 1 NADH and 1 CO2? So like, how many acetyl-CoAs is glucose composed of?
So, so the first step that glucose goes through is glycolysis. If you can see on here, each glucose makes 2 pyruvates. So there's 6 carbons in a single glucose molecule, and there's 3 carbons in a single pyruvate. So glucose makes 2 pyruvate through glycolysis. And once you have 2 3-carbon pyruvates, then it goes to the pyruvate dehydrogenase complex and makes 2 acetyl-CoAs. Does that help?
Yes, thank you so much.
You're welcome. I can't see your name, but and then last thing is that there is a way to regulate these complexes. So most enzymes are highly regulated based on the body's needs. And there's this enzyme pair called pyruvate dehydrogenase phosphatase and then pyruvate dehydrogenase kinase. So if you guys remember your like enzyme types, kinases use ATP to add a phosphoryl group onto something, and then phosphatases remove phosphoryl groups. So in this instance, a phosphorylated pyruvate dehydrogenase complex is inactive. And so phosphorylation occurs to deactivate pyruvate dehydrogenase complex in scenarios when there's high ratios of acetyl-CoA in the body, there's high ratios of NADH, and high ratios of ATP. So if you look at why that would be, you can see that when there's too much, this is feedback regulation, if that's a term that's familiar to you. Basically, when you have too much of the product, the body has mechanisms to slow down that reaction because you don't have a need for that anymore because you already have a lot of product. So acetyl-CoA and NADH are both products of that reaction. So when you have those two products in high amounts, the body will know to stop the activity or slow down the activity of the dehydrogenase complex because we already are satisfied in how much of these products we have. And then ATP is the ultimate goal of aerobic respiration. So you're trying to produce energy. So when you have a high amount of energy, you don't need to go through aerobic respiration as much. So it's another form of feedback regulation. And then once you have an inactive form, pyruvate dehydrogenase phosphatase is able to remove that phosphoryl group so that it becomes active once again. And this can happen in any different types of processes where you need a lot of cellular energy. So the example that's given here is that when there's calcium signaling, which can indicate energy demands by the cell. So whenever we don't need to know a lot of the detail of like the specific signals or the specific reasons why that would happen. Just know that this is one possibility. So mostly just know that phosphorylated pyruvate dehydrogenase complex is inactive, and a unphosphorylated or regular pyruvate dehydrogenase complex is an active form. And know that the inactivation is caused by high acetyl-CoA ratios, high NADH ratios, and high ATP ratios. I'll just take a moment here before we move on to the citric acid cycle if anyone wants me to repeat anything or clarify anything. Okay. So we talked about how glycolysis, we ended with pyruvate, and with the pyruvate dehydrogenase complex, we were able to make our acetyl-CoA. So the next step is that this acetyl-CoA enters the Krebs cycle. And I know this looks kind of intimidating right now, especially if it's something that you haven't studied a lot before. And I will warn you that this whole topic session is very memorization heavy. But that's just kind of the nature of these things. These are usually like pretty high yield topics. Like you at least get like one or two questions on this, on either the citric acid cycle, pyruvate dehydrogenase complex, or the electron transport chain. I would almost guarantee that you'll at least get a single question on any of these topics on like any MCAT. So it's really worth like spending a lot of time to kind of get these down. Um, I'll talk about this as I go through each step. You don't necessarily need to memorize each structure, but you would need to memorize each substrate in each step, each enzyme, and then what steps produce NADH and FADH2, GTP, and yeah. So you need to basically know a lot of information here. The only thing that you don't need to memorize completely is the structures. So anyway, sorry for that. But so back to this. So we have that acetyl-CoA generated from the previous step of pyruvate dehydrogenase complex. Now we enter this. So just like the pyruvate dehydrogenase complex, which is located in the matrix, citric acid cycle is also in the matrix. And just as an overview before we get into it, so we have 3 NADH produced, 1 GTP, and 1 FADH2 produced per 1 acetyl-CoA. So if you remember, we had this discussion about how 1 glucose yields 2 acetyl-CoA. So for 1 glucose, this would be 6 NADH, 2 GTP, and 2 FADH2. And if you guys aren't familiar with what NADH and FADH2 are, so these are structures that are very high energy because they carry high energy electrons, and they are used later on in the electron transport chain to produce energy. So the citric acid cycle is really about like gathering up these NADH and FADH2s. And then GTP is also a molecule that's high energy, but it's able to make ATP right away as opposed to being used up in the electron transport chain. And this is not a high energy electron carrier. This is just another form of energy. So guys, just take a look at this picture before we move on to the next step. Just take note that here's your 1 NADH. I'll start off this way. So you start off with acetyl-CoA and citrate. They form together, have a bunch of changes. Here's your first NADH forming. Here's your second. Here's your GTP. Your first and only FADH2, and your third NADH. So just 1 GTP, 1 FADH2, and then 3 different NADHs. Okay, so here's your first step. You're going from oxaloacetate, which is just a substrate that's a form that's available in the cell, and it combines with your acetyl-CoA to form this citrate. And this is what the citric acid cycle is named for. This is an irreversible step, which means that it's involved in rate regulation. And there's a slide about that later, but that's what's coming. And then the type of enzyme this is a ligase. So a ligase is an enzyme that takes 2 smaller molecules together and puts them together into a bigger molecule. And then the red box I just highlighted to show you where the changes are. So this kind of like that 4th carbon of the original oxaloacetate, it goes here. And this double bond O on the carbon becomes an OH. I forget like the name of the Orgo reaction this is, but I think it's aldol condensation maybe. I'm not entirely sure, but so those are the changes that are happening. And then here's your 2 carbons from your acetyl-CoA. Your CoA and then SH also leave, so you're left with these 2 carbons. And then your enzyme is citrate synthase. So fairly easy to remember because you're making citrate. And then your next step, once you have your citrate forming, this goes through a reversible isomerase reaction to make isocitrate. So again, kind of like intuitive, isocitrate from citrate shows you that something is just changing where it is to become an isomer. So like actually understanding the names is easier and more productive than like memorizing the structure. So that's why I keep emphasizing that you don't have to know the structure, just understand like what the changes are happening. And the enzyme, this is one of the harder names to remember, but it's a conatase. And that's because there's like an intermediate here. I think that's called something similar, a con. I don't know, but the enzyme is named for that intermediate between citrate and isocitrate.
Could you explain the isomerase the enzyme type, the isomerase, like that's occurring?
Yeah, so this is an isomerization reaction. So, do you need a refresher on what an isomer is, or is that not what you're asking?
Oh, that's what I'm asking.
Okay, yeah. So an isomer is something that has the same chemical formula. So it'll be like, so it has the same number of carbon, same number of hydrogen, same number of oxygen. The only thing that's different about it is like where the atoms are located. So if you see what I drew the boxes around, it shows you that this OH group just changed where it's located on the molecule. So these are what this is an isocitrate is just an isomer of citrate. It's composed of the same things, but the location of that hydroxyl group, the OH group, is different. So that's all that's changing in this reaction. And that's all that enzyme is doing. So conatate is an isomerase that's making an isomer of citrate.
Thank you.
You're welcome. And then step 3, so right now we're at isocitrate. Um, Here comes like a really common reaction type in the citric acid cycle. This is called a oxidative decarboxylation. So if you remember, this is the same type of reaction that we have in the pyruvate dehydrogenase complex. You lose a carbon as a carbon dioxide, and then this one OH group gets, well, this doesn't happen in the pyruvate dehydrogenase complex, but you have an oxidation where this OH group becomes a carbonyl group. So again, it's a redox reaction. So if your isocitrate is getting oxidized into what's called alpha ketoglutarate, then what needs to happen is that you need a molecule that's going to get reduced, and that molecule that you get is NAD plus. So this is where your first NAD plus, NADH plus form. Sorry, you start with an NAD plus, and you form an NADH, and then along with that, you also form a proton H plus. But you need to worry about this less, except to know that when you're making an NAD plus and you get an NADH in the citric acid cycle, what you also get is an H plus. And then also like a fairly easy enzyme name to remember, it's called an isocitrate. So your substrate's name, dehydrogenase. So you're removing these two hydrogens so that H that's on the NADH as well as the H in the proton also come from this original isocitrate molecule. So that's a way to remember it. So in summary, remember that it going from isocitrate to alpha ketoglutarate, you lose a carbon dioxide, form NADH, because NAD plus needs to get reduced if isocitrate is going to get oxidized. And this is a oxidative decarboxylation reaction. Again, it's a oxidoreductase enzyme, just like the pyruvate dehydrogenase complex enzyme. This step is pretty important because it is the rate limiting step of the citric acid cycle. And then it's another irreversible step, which means it controls the rate of the reaction. And there's more on that later, but the next step, so we're ending up at alpha ketoglutarate right now. The changes seem a little bit intimidating by the way I circled a lot of things, but it's very similar to the last slide, the last reaction that we just saw. So again, it's an oxidative decarboxylation. This carbon you lose as a carbon dioxide. This one that's already a carbonyl now, you make a carboxyl group out of it, so another oxidation. And then the only interesting part about this reaction from the last one is that you have this CoA that enters to form this group on the last carbon down here. So this product that forms is called a succinyl CoA. Again, so pretty easy enzyme name to remember, alpha ketoglutarate. So the substrate's name, dehydrogenase, because you get these hydrogens from the original substrate. The only thing is this is called a alpha ketoglutarate dehydrogenase complex. So again, same thing, if your alpha ketoglutarate is getting oxidized here, then NADH plus needs to get reduced. NAD plus needs to get reduced into NADH. So here's your other NADH. And then you have your CoA getting in to form succinyl CoA. But very similar to the last reaction overall.
I'm sorry. For step 4, is that when we produced our second NADH?
Sorry, say that again?
For step 4, is that when we produced our second NADH? Because I know you said in the beginning it's important to like know when.
Yes. No, go ahead. Sorry.
No, I was done.
Okay. Yeah, this is your second NADH. So here's your first one in step 3. Here's your fourth one. Step 3, first NADH. Step 4, second NADH.
Thank you.
You're welcome. Okay, so now we're at succinyl CoA. This step is a little bit interesting because it's different than what we've seen so far. So you have your CoA that just came on in the previous step, it leaves, and your like thiol group, I think this is, it changes to become a carboxyl again. And so this reaction, what it does is it forms GTP from GDP and in a inorganic phosphoryl group. And this part, it's not like a direct portion of the reaction. What it's showing you is that this GTP that's produced as a result of this reaction donates its high energy phosphoryl group to make ATP from ADP. So let me just say that again because I think I made that confusing. So what this reaction directly makes is GTP from GDP, but then the way that the cell is able to use that GTP is by making ATP from the GTP. And then this enzyme might be a little bit confusing, but it's not too bad. It's called a succinyl CoA synthase because it's named for the reverse reaction. So it's a ligase in the way it's classified because it's supposed to make succinyl CoA from succinate and CoA in the reverse reaction. But here it functions as a lyase, so something that's breaking a bigger molecule into two smaller molecules. Yeah, so this succinyl CoA synthase step is called substrate level phosphorylation in terms of the fact that GTP is making ATP directly, so you're not going through the electron transport chain, you're doing substrate level phosphorylation. And substrate level phosphorylation is also seen in a couple steps of the glycolysis process, so that's might be something that's more familiar. Okay, 6th step, we have 2 more steps, so just hang in there. Now we're at succinate, and from here, you form what's called fumarate. So if you see the differences that are happening, you have a regular, like saturated 2 carbons, and then you make a double bond from it. So this is, again, a different type of oxidation, but it's an oxidation anyway. So when you have your succinate that's being oxidized, something else needs to be reduced. Usually, we've been talking about so far, it would be the NAD+. In this case, because it's a double bond that's forming, you would have an FAD that's going to get reduced into an FADH2. So like NADH, this is another high energy electron carrier. And the clue to deciding when it's an NADH that's going to form when a substrate is getting oxidized or FADH2 is just that when it's a double bond, it's most likely going to be an FADH2. Other than that, this enzyme is called succinate dehydrogenase. So similar to isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. Let me see. So again, oxidoreductase. An interesting part about succinate dehydrogenase is that it's also complex 2 in the electron transport cycle. So it's part of both the citric acid cycle and the electron transport chain. And I put this specifically on here because in my practice, like AMC material and practice exams, I feel like I've seen questions that are like, which of the following enzymes are involved in both the citric acid cycle and the electron transport chain, and the only one that would be true for is succinate dehydrogenase. So next step, this is the second to the last step. So you have a double bond fumarate here. This is a hydration reaction. You have a water molecule that's going to join in to just add a hydroxyl group and then a hydrogen across the double bond. And then the enzyme name isn't too bad. It's just fumarase, similar to fumarate. And the name of the product that's formed is a malate. So this is the last step. So we talked about how oxaloacetate adds on to acetyl-CoA to form the first step of citrate. So to regenerate that oxaloacetate, because this is a citric acid cycle, so that cyclical form comes from starting with oxaloacetate and then ending with oxaloacetate. So anyway, all that's going on here is that you have an OH group that's going to be oxidized to a carbonyl group. So similar to what we talked before about if your malate is going to get oxidized, your NAD plus needs to get reduced. So this is your third and final NADH that's going to form. And similar to those steps which formed NADHs, this is going to be named as a dehydrogenase. So it's just malate, which is the substrate's name, and then dehydrogenase. And again, the product is called an oxaloacetate. So again, like very memorization heavy. Couple tricks to remember is that when it's a dehydrogenase, it's going to be a dehydrogenase. Let me find, here's the summary, and then I'll go back to the question. So you can see that I've highlighted where the NADHs form. So NADH, 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 and then FADH2. So anytime that these high energy electron carriers are forming, that enzyme is just going to be a dehydrogenase, and then in front of it is just going to be the substrate's name. So dehydrogenase here, dehydrogenase here, dehydrogenase here, dehydrogenase here, and then it's just going to be the substrate's name, isocitrate in step 3, alpha ketoglutarate in step 4, succinate in step 6, and malate in step 8. So that covers you 4 of the 8 steps. So really need to work hard on memorizing the other ones, but the other ones aren't so bad either. So like the first step is just citrate synthase because you're forming citrate. This one again is a little bit harder, aconitase, but it's not a particularly hard step to memorize because it's just isomerization. So at least for that step, all you have to worry about is like focusing on remembering the enzyme's name. Cover this. So step 5, again, might be a little bit harder to remember because it doesn't follow the dehydrogenase pattern, but you're just, and then it's kind of confusing because you're not synthesizing succinyl-CoA, but you have to remember that it's named for the reverse reaction. So it's just succinyl-CoA synthase. Just remember that the enzyme is actually named for the reverse reaction, and even though the enzyme is called succinyl-CoA synthase, you're going to be losing that CoA to form succinate. And then also remember that you have a GTP that's formed in that reaction, in that step, sorry. And then lastly, you have fumarate and malate, kind of easy to remember, but so it's just fumarase because the substrate is fumarate. And then just know that these two steps form carbon dioxide. So I have one question. Oh, I guess you would see it in the question. I've seen the answer, but I'll just quickly put a poll up here. This kind of like a harder question because I just took you through the steps, but try to see if you can work out based on what we talked about what the middle step is. So this is one step that we talked about, and this is another step. So this is asking you what's in between these two substrates. What is the intermediate? So note the changes, like you're forming an OH. Think about like from your organic chemistry background, like how is it possible to add an OH just onto an alkane? Like you can't, like what is that middle compound that you need to form to be able to add on a hydroxyl group? And I know this is tricky. You probably don't memorize, like have an understanding of these compounds yet, but just see if we can remember. And like I know I said that you don't have to memorize the structures, and that's mostly true, but there might be one or two questions like this, and you can work it out by like not memorizing the structures. So I'll take you through the reasoning. I got this question out of the AMC bank, and I was surprised that when I found it, I actually got this right. Because after like not studying it for a while, I thought like that was a pretty tricky question, but I think I just worked through it as opposed to like actually remembering the structures. Okay, I'll just stop it in like 5 seconds. I'll show you the results. It's pretty evenly split, which is totally fine, but B is the correct answer. So here is what I highlighted, like I was telling you, like this is a single bond, and then now you have an alcohol forming. So the way I thought about it is like you probably need a double bond here to be able to add on that hydroxyl group, and that's true. So fumarate is the middle, the intermediate between these two substrates. And I think I was able to reason that um, fumarate forms as a result of succinate dehydrogenase, and succinate dehydrogenase produces FADH2. And then if you remember, we talked about how FADH2 results when you're forming a double bond. So some of it is a little bit of like luck based on like which compounds are in your options, but you have to work hard on like reasoning those out. It's a bit easier because this one I think was possible because fumarate is the only um, one only compound in any of these steps that has a double bond. So I think they'll typically give you ones where you don't necessarily need to know the exact structure, but just know like its features that are unique from other compounds. So yeah, I mean, it's mostly memorization, but understanding it will save you some memorization. And then another question. So again, this is a memorization question. I don't know if you guys just want to skip it, but I'll just put it out there for like a minute to see. So basically what this is asking you to do is that it's limiting you to a specific part of the citric acid cycle. So if you remember, the citric acid cycle starts off with citrate, but here it's telling you don't start off there, go all the way to alpha ketoglutarate and then end up at the end, oxaloacetate. So it's kind of asking you between citrate and alpha ketoglutarate, if we get rid of that, how many electron carriers are formed there that we'll miss out on if we start off at alpha ketoglutarate. And then, so I also like this question because it doesn't ask you NADH or FADH2 correctly. You have to understand what they actually are, and they're these reduced electron carriers. So I kept talking about how because those substrates were getting oxidized, you were forming these NADH and FADH because they're going to get reduced to compensate for the other substrates that are getting oxidized. So you should know that they're in the reduced form and they're carrying these high energy electrons. So in this question, it's just asking you how many molecules of NADH and FADH are formed between alpha ketoglutarate and oxaloacetate. Okay, I'll just end it in like 10 seconds. So most people picked B. Should have the answer. It's actually 3. I don't know if you got confused between like NADH or an FADH2 or just didn't remember, which is fine. But so if you look here, if we start off at alpha ketoglutarate, you did miss an NADH up here if you skip through these steps. So in total, we form 4 in general. So we form 3 NADH and 1 FADH2. So if you exclude these steps, you're only missing 1. So from 4 total, so 3 NADH and 1 FADH2, you normally have 4 electron carriers that are formed in the citric acid cycle. If you just take out 1, you're left with 3. So here's your first one. If you start off at alpha ketoglutarate, here's 1 NADH, 1 FADH2, and your second NADH. So 2 NADHs and then 1 FADH2. And GTP, remember, is not an electron carrier. Okay. Is there a question? Okay. So here's just a general summary of the citric acid cycle. So this is balanced like 1-to-1. So if you look at the acetyl-CoA, it's what becomes of it is that you have, remember those 2 carbon dioxides that are released in step 3 and 4. Those are what comes of that acetyl-CoA. And then in 3 different steps, you have NADH plus being reduced to NADH. And then with NADH forming, you also release 3 protons, so that's where these protons are coming from. And 1 FAD is reduced to 1 FADH2, and then GDP and inorganic phosphate in one step form GTP, and then this is the remnants of your acetyl-CoA that's not released as a carbon dioxide. So remember that these are for like 1 acetyl-CoA. So don't fall into the trap when they ask you like how many NADH and FADH2 are formed in the citric acid cycle for 1 glucose. Remember that each 1 glucose equals 2 acetyl-CoA. So you have to multiply all of this by 2. So 2 carbons are completely oxidized to 2 CO2. This is kind of like a fact that you don't have to worry about too much, but there's 8 hydrogen atoms that are released in the form of reduced cofactors. So that's where you get those NADH, and then this proton, and then FADH2 form. This is where like why all of those enzymes are called dehydrogenases, because you're removing hydrogen atoms. And then this is just to show you that when you generate these electron carriers, what is going to become of them is that you're going to make ATP of them. So each NADH makes 2.5 ATP, and then 1 GTP just makes 1 ATP, and then 1 FADH2 makes 1.5 ATP. So per each acetyl-CoA, that's 10 ATP, and then so 20 ATP forms in the per glucose when you're talking about just in the the citric acid cycle. And then this is a mnemonic from Kaplan to remember the substrates, if that's a way that helps you remember it. But just like practicing a lot with it will also kind of let you remember the order. So either way is fine. Just try different things, and you'll. It's kind of frustrating. It was frustrating for me to keep reviewing this, and I would always forget. But that's just kind of the nature of this stuff. It is very memorization heavy, so just look at it often. So I'm sorry, I don't know how to say your name, but so 2-acetyl-CoAs are not produced in the citric acid cycle. So remember that acetyl-CoAs are produced in the pyruvate dehydrogenase complex, and the citric acid cycle uses up these acetyl-CoAs. What I said in terms of this 2-acetyl-CoAs is that your starting energy molecule, so the thing that you're metabolizing in these entire process is glucose. So that I was saying that from one glucose, you generate 2-acetyl-CoAs through glycolysis and through the pyruvate dehydrogenase complex. So the citric acid cycle uses up that acetyl-CoA. It doesn't produce it. You're welcome. And then regulation questions are also very important to know and come up often. So I briefly mentioned as we were going through the steps that there are certain enzymes and in certain steps of the citric acid cycle that regulate the rate of the citric acid cycle. And these are citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase complex. And out of these 3 steps, the most important one is isocitrate dehydrogenase because this is the rate limiting enzyme. It determines the rate for the rest of the citric acid cycle. And this is just a matter of memorization again. I don't really have good ways to keep these in mind. And so when you have regulation, that means that there's certain things that inhibit this step and certain things that activate it. So before we look at the individual steps, I wrote up here that ATP is an inhibitor of all 3, which makes sense because the whole goal of aerobic respiration is to form ATP. So when you have a lot of ATP, you don't need this cycle to proceed. So you can save cellular resources by slowing down the cycle when you already have a lot of ATP. On the other hand, ADP is an activator of all 3. So when you have high ADP concentrations, it means you have less ATP concentrations. So you need to tell the citric acid cycle to speed up. And then NADH is also an inhibitor of all 3. So similar rationale, like the citric acid cycle produces NADH. So if you have a lot of that product, it's feedback regulation that the cycle can slow down. The tricky part is that NAD plus is only an activator of the isocitrate dehydrogenase. And you can kind of think of this by remembering that isocitrate dehydrogenase is a rate limiting enzyme. So it's a little bit more, I don't know, special, I guess. So while the other 2 are only activated by ADP, you can remember that isocitrate dehydrogenase is a little bit more important and is activated by ADP and NAD+. So now to look at the other ones that we haven't covered. I said ATP and ADH for all of them. Citrate is another activator of citrate synthase. So when you have a lot of reactant, you want to use it up. So you want to speed up that reaction. So that's kind of something that makes sense to us through Le Chatelier's principle, even though that's for equilibrium and this is looking at rate. It's kind of similar logic that you can use to think about it. And this one is a little bit harder to remember, but succinyl-CoA is also an activator. Not entirely sure what the reasoning for that is, but could just memorize it. So these we've covered. The only other one we haven't covered down here is succinyl-CoA is an inhibitor of the alpha ketoglutarate dehydrogenase complex. And if you look up here, succinyl-CoA is a product of this step. So it makes sense. It's again just feedback regulation. When you have a lot of the product, this step can slow down. Oh my god, I do not remember if ANP is an activator of this reaction. I think this table is comprehensive and I think it's not, but I'm actually not sure. Hope if anyone doesn't mind if I just look it up real quick. Thank you for asking that question. Yeah, I guess it says like here that it's the ratio of ATP to ADP and ANP. So um, I think it might be. I haven't seen it specifically come up in my review, but I know it specifically is like a regulator of a couple steps in glycolysis, but um, I guess I wouldn't worry about it too much. Sorry for not knowing the details of that. Yeah, it's kind of like similar to ADP. So while ATP is like that high energy molecule that you can use, ADP and AMP are like the used up, like a result of like using up ADP. So finally, we have the electron transport chain. This is located in the cristae. So this is the inner membrane. So we're moving out from the matrix up into the inner membrane. And an electron transport chain isn't used just for aerobic respiration. So prokaryotes also use different types of electron transport chains. So I just included it in here that like when prokaryotes need electron transport chain because they don't have mitochondria, this is located in their cell membranes instead. Just like a fact that might show up in like a discrete question somewhere. So what the electron transport chain is, it's basically a set of redox reactions to harvest energy from those high energy electron carriers that are NADH and FADH2, which we created in the citric acid cycle. So whatever you generate in the previous step, so we created acetyl-CoA in the pyruvate dehydrogenase complex, we use that up in the citric acid cycle. Now we made NADH and FADH2 in the citric acid cycle, we need to use that up through the electron transport chain. And here's just an overview. It's probably confusing and not so helpful right now, but I'll again go through it step by step, complex by complex, and hopefully that'll help. So here's our first complex. It's called NADH ubiquinone oxidoreductase. More commonly, it's just called NADH dehydrogenase. So again, there's a dehydrogenase word, which means that it's just going to remove that hydrogen off of NADH to make NAD plus. So NADH carries these two electrons to FMN, which is like a cofactor of this enzyme complex. So it basically is passing on these high energy electrons to something else. And then FMN then carries these electrons to ubiquinone, which is represented by Q here, and reduces that to QH2. And then QH2, which is called ubiquinol, carries these two electrons into the next complex, but something that's important to remember is that complex one makes ubiquinol, but the ubiquinol doesn't go to complex 2 right away. It goes to complex 3. And here's just a summary equation. So when you have these electrons being passed down, that energy also allows you to pump protons from the matrix into the intermembrane space, and this will become important later on when you're actually making ATP. So right now we're harvesting energy from the electrons to make a proton gradient across the inner membrane of the mitochondria. So the goal of these 4 complexes is to push to actively pump protons. I shouldn't say actively, you're not using up ATP, but you're using the energy from these electrons to pump protons into the intermembrane space so that there's a difference in the concentration of protons across the membrane. See, there's a question in the chat. Is a electron transport chain same thing as oxidative phosphorylation? Yeah, kind of. So oxidative phosphorylation refers to the fact that you're using redox reactions to make ATP. So that's used more of a term to differentiate from substrate level phosphorylation, which is where you just have an inorganic phosphate phosphoryl group from a different source to make ATP. So electron transport chain is a component of oxidative phosphorylation. You're welcome. So I just briefly included here what ubiquinone and ubiquinol look like. Weirdly saw this come up on a question, so I just thought I'd throw it in there. It's like not too difficult to memorize. So here's the oxidized version of it. So this is what it starts out as. And then once NADH passes on these two electrons, it becomes ubiquinol. So this is the reduced version. So this is a general kenone structure, if you guys remember what a kenone is from Orgo, but instead of having like 3 delocalized pair of electrons, it just has 2, and then these 2 carbonyl groups. And then once it gets reduced, it forms 2 hydroxyl groups here, and then like a regular aromatic ring. Yeah, so I guess just like know the general structure that ubiquinone is 2 carbonyl groups like this, and then just 2 pairs of delocalized electrons, whereas ubiquinol is 2 hydroxyl groups like this, and then like a regular benzene ring. So the next complex is the succinate ubiquinone oxidoreductase complex, more often known as succinate dehydrogenase. So that dehydrogenase gives you a hint that you're taking off. Well, actually, this is what I was talking about earlier when we were talking about the citric acid cycle. This is an enzyme that's also functional in the citric acid cycle. So it reverts to the fact that you were oxidizing that succinate. So you form that fumarate and generated FADH2 in the electron transport chain. Now, what's going to happen to that FADH2 is that it's going to get reduced back to FADH as it passes on the 2 electrons from itself onto these F iron sulfur centers, which are again a different type of cofactors in the succinate dehydrogenase complex. And then once these centers get it, they're going to pass on that electron again to ubiquinone to make ubiquinol. So same thing, same general process as complex 1. So complex 1 is making ubiquinol from electrons from NADH. Complex 2 makes ubiquinol from electrons from FADH2. And an important thing to note about this complex is that complex 1, 3, and 4 all pump out protons, which is really important for making ATP later on, but complex 2 actually does not. So, okay, so now we're at complex 3. So at this point, we have a lot of ubiquinol from both complex 1 and complex 2. So now what that does is that ubiquinol gets reduced back to ubiquinone as it passes the electrons again to this heme protein called cytochrome C. So it's interesting to note that in complex 1 and 2, you pass on 2 electrons at a time. That's not the case for cytochrome C because the heme group only alternates between the ferrous, so the +2 form and +3 form. So it can only take on 1 electron at a time. So the ferrous, the +2 form, is the oxidized form, and the ferrous is the reduced form, and the ferric is the oxidized form. So reduced, so the one that you start off with before it has any electrons is cytochrome C oxidized form, so that would be the ferric +3 form. And once it gets that electron from ubiquinol, it becomes the reduced form, which is the ferrous +2 form. Then this is called the Q cycle when it's going back from ubiquinol back to ubiquinone, and then it needs the help of a second ubiquinol to go back to ubiquinone actually, because the first one donates both electrons and becomes a radical semi-kenone, which is up here, but you need a second ubiquinol to help that radical get back into the ubiquinone form. It's not something that you need to know the details of. It's just a little bit more complicated. Just know that the Q cycle takes place in complex 3 for the purpose of regenerating ubiquinone. And again, you pump out 4 protons, similar to complex 1. Yeah, complex 1 was also 4. So 4 protons in complex 1, 4 protons in complex 2. And then the last complex, sorry for going a little bit over time. We're mostly done. I have like 5 slides left, I think. So this is called cytochrome C reductase. So previous one, if you remember, was called cytochrome C. Oh, sorry, this should be cytochrome C oxidase. No, okay, wait. So this is cytochrome C reductase because you're using those electrons to make oxidase, the oxidized form of cytochrome C into reduced form of cytochrome C. So this last one should be cytochrome C oxidase. And that is because the electrons that the cytochrome C has now are going to be passed on to your final electron acceptor. So the cytochrome C is going to get oxidized again. And your final electron acceptor, we talked about a little bit before, is oxygen. So that's where you get that water that's formed. So this is a step where that really, a lot of people know why aerobic respiration is aerobic. You need that oxygen in aerobic conditions to be able to accept that electrons to form water. So here's a step where you generate water. Don't really need to know how many waters, whatever. It's not super important. What you need to know that, again, you have 4 more protons that are generated into the intermembrane space. So again, more protons to form that proton gradient that will use to make ATP. So you pump out 10 protons for each NADH. So 4 in complex 1. Okay, so I misspoke earlier. So it's only 2 protons in complex 3. And then, no, sorry, wait. Like I'm getting mixed up with the protons. It's not like super important to remember like which step has which protons, but just know the total amount. So 4 in complex 1, 4 in complex 3, and 2 in complex 4. So 10 total, and then 6 for FADH2 because if you remember, FADH2 skips complex 1 and goes directly into complex 2. So because you're missing out these 4, it's only 6. So 4 from here and then 2 from here. And then I have here that NADH is highest in energy and oxygen 2 is low, or oxygen is lowest in energy. So as you fall through these complexes, the electrons become like less and less lower and lower in energy because naturally as you pass them on, they lose some energy. So I think that's what's referring to. And then another thing to mention is that as you go through these complexes, each subsequent compound has a higher affinity for electrons. So oxygen has the most affinity for electrons because it's the final electron acceptor, and NADH has the least because it's the first one to give up the electrons right away. So here's a question. Just the poll. Should be pretty quick because I mentioned it directly. I'll give you 2 more seconds. So yes, almost everyone picked complex 2, which is correct. If that person who got it wrong wants me to explain something, please speak up or put it in the chat, but I'll just take you back to the overall picture here. So you have that complex 1, complex 3, all pump out protons into the intermembrane space, but complex 2 is that one that harvests those electrons from FAH2, and it does not. And complex 2, if you remember, is also part of the citric acid cycle as well. So just another quick question. Which of the following components of the electron transport chain has the greatest attraction for electrons? So that's the whole basis for the electron transport chain. You're moving along electrons, so how is that happening? Okay, let's give it 10 more seconds. Okay, so the majority did pick oxygen, which is correct. I think NAD plus is a little bit confusing because it is the oxidized form, so it does attract electrons. But overall, like in terms of the electron transport chain, your final acceptor is still oxygen. So if you think about it, oxygen definitely accepts electrons from cytochrome C, so it's definitely higher affinity than cytochrome C. But cytochromes, so in that logic, cytochrome C accepts electrons from ubiquinol, which is so it's higher than ubiquinol, but then ubiquinol accepts energy electrons from FADH and NADH2. So using that logic, you can see how oxygen has the most highest affinity. So the only one that's a little bit confusing here is NAD plus, but just as a general rule of thumb, in the electron transport chain, oxygen will have the highest affinity for electrons. Okay. I'll go through this as quickly as possible, I'm sorry. So we talked about how you get that proton gradient established by the complex 1, 3, and 4 creating that gradient. So the purpose of generating that gradient is so that they will flow down that gradient through osmosis. So chemiosmosis is just the same thing as osmosis. And when it flows down through these enzymes called ATP synthase, which do exactly what it sounds like. They use that proton gradient, so the force of that protons moving down that enzyme to convert ADP into ATP. And this ATP formation is regulated by the ATP concentration as well. So earlier someone asked what is oxidative phosphorylation? Is this the same thing? So this is the oxidative phosphorylation from it. So these protons we generated in the intermembrane space by the use of the redox reaction in the electron transport chain. And here's where we use that that proton gradient generated by those redox reactions to phosphorylate our ADP into ATP. Here's just a diagram that you can kind of refer to on your own time. It's generally good to know like where these go. I would know like that the catalytic sites are located in the matrix, and that there's a rotor that by the protons flowing through them, it moves them, and that's the mechanism of forming the ATP. Yeah, just you can look at it. It's not too important. Just know there's like 2 things I mentioned. And here's a summary of like how many ATPs you make overall. So glycolysis, you make 2 NADHs and then 2 net ATPs. Pyruvate dehydrogenase complex, 2 NADH per glucose, because it's 1 per acetyl-CoA. Citric acid cycle, so it's 3, 1, 1 per acetyl-CoA, so it's 6, 2, 2 per glucose. Once you add them all up, you have 10 NADH, 2 FADH2, 4 ATP, and then so it's 2.5 ATP. So this is just a number that you can memorize. 2 ATP per 1 NADH, 1.5 ATP per FADH2, and then 2 ATP just, sorry, this should be 4. 4 ATP. So once you add all that up, you get like approximately 32 ATP. Sorry, let me just fix that so I don't forget. And then I think this is the last question. If thermogenin, a protein present in the inner membrane of the mitochondria of brown adipose tissue, uncouples oxidative phosphorylation and ATP production, which of the following will occur? So let me launch the poll again. And I'm sorry for keeping you guys a little bit late. I thought I planned this today to go on time, but I'll keep working on it. I'll give you 15 seconds. So this is fine. So we recognize that somehow the ATP formation will be lowered, and the answer is that no ATP will form. So this is called completely uncoupling it. Yeah, that's correct. So thermogenesis, so instead of making ATP by that protons flowing down the ATP synthase, what thermogenin is like another protein that sits in the inner mitochondrial membranes. So the protons can instead flow through them. And what that enzyme does, it generates heat. So it completely diverts ATP from that from the ATP synthase to thermogenin. And I think like this is a bit confusing the way I think I probably should have not put option A. When you're on the MCAT, it'll be pretty clear. Like you, I think it'll be easy. Like I didn't mean to confuse you with giving you the less ATP option, but I see how you might think that is true. But on the original question, I think it was just no ATP will form. So your choices will at least be more clear. And that's the end of it. So the only thing we wanted to tell you is that we're working on establishing this one-on-one tutoring. We want to try it out free at first and see if there's any interest in getting paid tutoring. But we request that if you're interested in it, something that you think you could benefit from, this interest form is something that you could fill out. And I'll include this in the post-session email. But please fill that out if you're like seriously thinking about utilizing this service. We don't have a lot of interest right now, so we're trying to get a feel for like if this is like a doable like taking. And then any questions, as always, you can email us at that email. I'll send you a post-session email with the recording, the slides, and the interest form, as well as a post-session like evaluation of how I did. So if there's any suggestions that you can give me, anything that you thought I did well, we would appreciate that. So yeah, thank you for staying. I hope this was helpful. If there's any feedback you want to give to me right now, that would be great as well. You're welcome.